As I said at the beginning of this chapter, we really focus on what cells have in common as a way to understand them and to study their organelles and study basic cell structures. But in reality, cells vary widely depending on what function and role they play in an organism. And just to give you an image for this, these are skin cells, and these are loose skin cells, and you'll see them when you do this in the lab, in the experiment. If you were to look at those skin cells in place, they would be a little more like this. And in fact, sometimes when you do the experiment, when you look at your own cheek cells, you'll see little ribbons of this still. They stay connected to each other, and inside of your cheek, they'd be sitting on a layer of tissue called the extracellular matrix. So this is a very characteristic type of cell called epithelial cells. In contrast, this is a nerve cell or a neuron. And these cells don't hang out together like this, and they have this very spindly, spread out structure. And these differences in shape reflect their differences in functions. The skin cells are primarily providing structure and a barrier, whereas a nerve cell is transmitting messages down the nervous system. So we see differences in shape and size. Cells can also differ in their organelle composition. So if you think about a liver cell and compare it to a muscle cell, what is the role of the liver? Well, you probably know that it's involved in things like detoxification. For example, alcohol. Well, what organelle detoxifies alcohol? If you remember, it's the smooth ER, right? So you might expect that a liver cell would be rich in smooth ER. A muscle cell, it's not detoxifying. It's really primarily involved in movement. So you're going to see a lot of these filaments. Remember we said actin filaments and motor proteins. So you're going to see lots of that. And then the other thing is muscles use a lot of energy. So you're going to need something that processes energy. So you see a great deal of mitochondria in muscle cells as well. So you have a lot more mitochondria in muscle cells than liver cells, and you have a lot more smooth ER in liver cells than in muscle cells. So, and that's just an example, but depending on the cell type, it's going to have very different composition. Even to the point of not even having DNA in it. Some of you may know that red blood cells, for example, don't have DNA in them. They're so specialized to carry oxygen in our blood that they won't even have a nucleus. So in this example, hold on, let me go back to this picture here. In this example, this is a human skin cell and this is a human nerve cell. So this could be from the same exact person. That means their DNA is exactly the same. And I know that you've heard that DNA is the instructions for life, or your DNA is the instructions for you. So how does the DNA, that's the instructions, account for making this and this if they have the exact same DNA? Do you guys see what I'm saying? There's DNA here, and if it's coding for you, it's the same exact DNA as coding for this nerve cell. Well, the difference between the two cells in the same individual is really a difference in the protein makeup. So you can think of DNA as kind of like your big filing cabinet full of instructions on how to make an organism, right? But every cell is going to use different filing folders. So every cell gets the whole filing cabinet, but they'll only pull out some of the folders and only read some of the folders. So that way you might only get certain proteins. So if I think about my DNA, do really bad sketches here. My DNA is the whole filing cabinet. There's little handles for pulling out the filing cabinet. So we're talking about thousands of folders of information. And every cell is going to get that same filing cabinet, but if it's a liver cell, it will only read certain folders. And if it's a muscle cell, it reads different folders. So some the same, some different folders. Or it might read them more or less. This one might use a folder every single day. This one might only read a folder once a week or once a month. So from the same filing cabinet, if I use different folders, I will get different proteins. So I get different proteins in the cell depending on which of the filing folders get read. And again, this term for all of the proteins in a cell is the proteome. So the proteome and the proteomes between the two cells are different. And it will be the difference in proteins that fundamentally allows the cells to have different structures, different shapes, and different functions. So the last note here about the cell is that we've sort of gone through this laundry list of organelles, and to be honest, like that's not the funnest thing, right? And so I just want to remind you that the cell isn't just throwing in this grab bag of organelles and da-da, you have a working cell. The cell really only functions when there's an integration of all these different structures and organelles, and that integration really has to be inherited from the pre-existing cells. So the integration is so complex, we can't just mix it all together, stir it up, and get a cell. The integration and the structures are so complicated that they have to actually be inherited from pre-existing cells. So that sort of wraps up and connecting back to the cell theory. Every cell must come from pre-existing cells to get this integration of structures. And this is just an awesome picture that I love of an immune cell, and these are bacterial cells that it's attacking. So a very complex function being facilitated by complex shapes and a unique set of proteins that will allow this immune cell to hopefully keep this person from getting sick. And so that's it on the cells. And like I said, this is a preview for a lot of the rest of the semester, as I alluded to throughout the videos.